Hello everyone. Today I will show you how to use Cytoscape. Cytoscape is a wonderful tool to visualize networks. It also provides some basic uh, tools for analyzing networks. First, we need to download Cytoscape. Just search for Cytoscape download, and you'll get this page. And you can just download it, and the installation procedure is fairly easy. You just accept the license agreement and select next, and it will be finished. Now will take a lot of time to download this, so I'm not going to show you the download process. It's fairly easy, so you should be able to figure it out. Now after installing Cytoscape, you will be able to find it by searching in Windows search panel, and you just click here, and then you will see a window like this. So this is the window you get when you open Cytoscape for the first time. Here in this window you can see that there are several options to create a network. You can create an empty network, you can create a network, you can import a network from a file, you can construct a network from a network database, or you can just select some species, a specific network from here, and thereby you can get the network. Here, H sapiens means Homo sapiens, which means human beings. H cerevisiae means Baker's yeast. Mus musculus is mouse, so on and so forth. For the purpose of this tutorial, let's look at how we can create a network from network database first. So click here, open network database. Here you can see that there are several databases available. EPID, Mentha, BioGrid. BioGrid is one of the most comprehensive network database, and it shows the types of edges, types of interactions available in this database as well. Like BioGrid contains protein-protein interactions which are internally curated. It's not predicted. It's experimentally found, and it has been curated by experts. So it's a very reliable database. And then also there are some other databases. Let's look at some other databases like Intact. Intact contains protein-protein interaction as well as a small molecule protein interactions as well as nucleic acid protein interaction. So small molecules means some kind of molecule that with which a protein binds. It can be a drug molecule as well. And nucleic acid, you know about nucleic acid, the RNA and DNA. Proteins can bind with these kinds of nucleic acids and thereby perform some task. So Intact contains protein-protein interaction as well as small molecule protein interaction as well as nucleic acid protein interaction, so on and so forth. So now you see that you cannot select any of these databases. Why? Because it did not provide any list of gene here. You have to provide a list of gene here, and then click the search button. If the corresponding interactions for those genes are found in a database, then those databases will be shown in active font, and then you can select or deselect them. So let's write down some names of genes which I know about. Say LG5, LG6, LG3, LG12. These are some yeast genes. Let's search here, and you'll see that only top few databases contains interactions between these genes. So let's look at Mint, or maybe BioGrid. BioGrid is very comprehensive one, so let's select BioGrid. You can also select Mint, and I want to get only one network from this, from the merging of these two networks. By merging these two networks, that results from BioGrid and Mint respectively. So I'll select this option as well, automatic network merge. Then I'll just click import. As you can see, when I'm importing interactions from remote databases, it will take some time. Okay. Since there are only 4 genes here, it did not take much time, but if you provide hundreds of genes, it will take a lot of time and it will consume a lot of memory. So this is one downside of Cytoscape, that it consumes a lot of memory, but the kinds of visualization it provides is really awesome. That's why it is very popular. It is really popular among network analysts and researchers who work with networks. Now what it is saying networks created from the following databases are merged. So it is already merged, so I'll just close it, and I don't need this panel either. So let's just cancel. Okay, so what I am seeing here is the network created by merging these two merging the networks resulting from the two databases, namely BioGrid and Mint. And let's move this here. Let's maximize it. Okay. So you cannot see the names of any genes here, right? So what is going on? We can actually zoom it out by pressing this button, or just scrolling our mouse up, and that way we can get closer to the nodes, and then you will be able to see the names of all the genes. So as you can see that there are lots of nodes here, although we have provided the names of only 4 genes. So where are those 4 genes? So you can search your genes search button, and it also provides some kind of help, like YL star, search all columns for YL star, and name colon YL star means search name column for YL star. So YL star means what? YL star means any string that starts with YL, any word that starts with YL. So let's search for, okay, it should be capital LG star. Okay, here it is. So as you can see, this is I don't know, maybe LG 5. So let's see, this is LG 5. This is LG. Let's see it. You can move very quickly using this insert. So this is LG 6 here. So it is showing it in a different name. In biology, a gene can have multiple names. So I have used human readable labels, but they are using some other name for this gene for visualization. So when I provided only the names of only 4 genes, what happened is it searched for all the neighbors of each of these 4 genes, and it is showing the network created around each of these 4 genes. Also notice that when I searched for ALG6, only 3 gene names matched with ALG6, which are shown here and which are also highlighted by yellow color here. And also, oops, I zoomed in, I should zoom out. Also here, as well as here. So these genes are shown in different names, but all of them actually maps to the name ALG6. All of them map to the name ALG6. Okay. So can you quickly tell me how many connected components are there in this network? You already know about connected component, right? So connected component of a graph is a subgraph in which there is a path between every pair of nodes, right? So for example, this is a connected component. There is a path between every pair of nodes in this subgraph. This is another connected component. This is another connected component. This one. So on and so forth. So we can of course count them by hand, but that will be inefficient. Cytoscape actually provides a quick way to compute the number of connected components you have, and to do that you have to go to tools and select network analyzer, network analysis, and oh no, sorry, subnetwork creation, extract connected components. Okay, it quickly gives you summary of the connected components present in this network. So the largest connected components contain 306 genes. The second largest contains 105 genes, so on and so forth. And you can extract any of them. So let's extract the largest one, which is this. So what happened now? In the left panel, what you are saying, it is called the control panel. Let me close it. So this is called control panel, and if it is not visible, which may happen if this is selected, right, control panel. If it is not visible, just go to view and select show control panel, then it will be visible. In control panel, you see there are several tabs for network, style, select, etc. Now each bullet, top level bullet in this network tab corresponds to a collection of networks, which is automatically named as merged network. If I expand this bullet, I get merged network. So this was the initial network that we got from the BioGrid and Mint database, and this is the network we have created from the previous network by extracting the largest connected component present in it. So you can also see that there are 2 numbers beside each network. The current one, there are 2 numbers for the current network, which are 306 and 481. 306 means there are 306 nodes in this network and 481 edges in this network. If you go back to the previous network, it says 531, means there are 531 nodes and 731 edges. Okay. So let's go back to the largest connected component. Why choose largest? Typically this is what we do in computational systems biology, that we analyze the largest connected component of a cellular network. Cellular networks are often disconnected, meaning there are multiple connected components in them. But for us, the most interesting one of those connected components is the largest one, because that gives us most information, that contains most information. Okay, so now let's what we are doing, we are searching for ALGs. So let's search for ALG star. So these are all ALG genes here. Okay, now you can also search using some column name colon, some name like biogrid colon. What is this? 32938. 32938. Okay, so it is saying could not find any matches. So I believe I have to put escape sequence to match these things. Okay, still could not find. Oops, I made a mistake. Column should be after. Okay, now it matches. So yeah, so this is what I wanted to show you, that when you want to match with a certain column, when you want to have a certain value in a certain column, you put the column name here and then put a colon and then put the string that you are looking for. So as you can see that it is showing in this panel, it is showing only the information about this node, which has biogrid name 32938. And you can also do and of course you have to escape the third brackets or the square brackets by using slash, otherwise it will show an error which you have seen. If you want to search using the name column, so for example to find some gene which has name 32938, I have to put like this, and again only this gene is selected. So let's click somewhere else so that all the genes information are shown in this table again. So how about some genes whose name starts with 318? Let's see. So to search for genes whose name starts with 318, I have to write name colon 318 and then put a star and then press enter. So as you can see, the list becomes shorter, and all the gene names that appear here have name that start with 318. Okay, so we have seen the searching option, and we can of course expand it, zoom it, and we can zoom out to display all of the current network. We can actually detach this whole network, so that will be another window, another independent window where we can work. Okay. And so now this is a very small network, so there is not much complicacy here, but when the network becomes very large, then this kind of tools like moving from one part of the network to another is very useful. So this insert tool is very useful when the network is large and you want to um, zoom out as well as move from one part of the network to another. Okay, now we have talked about some network for which we already knew the names of the genes, but this may not be the case in general. Typically, we may not know the names of the genes. So what can we do? You can um, import some network file. So let's see how we can import some network file. To import some network file, you have to go to file menu, then select import, then network. Then there are three options: file, URL, public database. If we select public database, then we will get a window like this, where again we have to put the names of the genes around which you want to create a network. But if you already have a network file, then you want to select file, import network, then select file. Now there are already some sample network files available in Cytoscape. Where are they available? We typically install our program files in C program files, which I did as well. Under that, there is a directory called Cytoscape version 3.4, and under that directory there is a folder called sample data. In this folder, you can find many different data that can be imported into Cytoscape. So let's select this file, ist high quality.sif. So SIF is a network file format. Typically in this file there are three columns. First column indicates the first gene or first node in an edge. The third column indicates the second node in an edge, and this second column indicates the type of relation between those two nodes. Okay, without further ado, let's select this and open. And it is asking me where to put this network. We can create a new network collection, or we can put this under merge network. If we put it under merge network, it will be shown as a sub menu of merge network menu. So I don't want to do that, because this is a totally different network. So let's create a new network collection and select OK. Now this is a very large network as compared to the previous one. It has more than 3,000 nodes and almost 7,000 edges. So it looks really complex. And as you can see in this panel, this is the table panel. There are only two columns available: shared name and name of a gene. We did not see these other tabs earlier, so let's look at what the other tabs show. Age table shows the information about age. The PP means protein-protein interaction, which is a physical interaction, physical association between two proteins. Network table shows the name, shared name of networks, etc. Let's go back to node table. Now you can zoom in. You can zoom in this network to see the names of different genes. It looks like you have to zoom out a lot. Sorry, zoom in a lot. Okay, let's just select some node. It is not showing anything yet. Okay, so why it is not showing the names of the nodes? To investigate this, let's look at this tab, the Style tab. And if you go to Style tab, you see that this label menu here, there is some exclamation sign. Some kind of error has been happened. So the column from which it was reading the name of a label is human readable label, but there is no such column for our current network. For our current network, there are only two columns: name and shared name. So each column describes some information about the gene. So let's select name here, name for our label, and then suddenly all the labels of nodes have appeared. So since we have already come here, let's talk about the Style tab a little bit. So from this tab you can edit the style of nodes and edges and the network in general. So for example, here you are reading the label from a name column. You can also read it from shared name, which is probably the same thing. Let's keep it name. Okay, you can select a different value for a node which will appear as a label of every node, but we don't want to do that. You can change the label color. So currently it is black. You can make it red, for example. So all the label colors become red. And you can change the shape as well. The shape is currently round rectangle. You can select ellipse, like all of them become ellipses. Then you can change the size of the nodes. You can change height and width of the nodes, so on and so forth. Border, paint, border width. You can currently the border of each node is gray. You can change it to something like cyan. So this is just for changing a style and visualization purposes. So as you can recall that Cytoscape is mainly a visualization software, and so it provides a lot of tools to visualize a network as your choice, as you choose. And this types of visualizations are really important for publishing in good journals like Cell, Nature, Science, etc. So they are really keen to see good quality pictures. So that's why these kinds of software is really important for research applications. Okay, so now we have shown how to search different nodes and how to change the styles of the network. Let's take a tour of these different menus. So from File menu, you can export networks, you can import networks, etc. From the Edit menu, you can do Ctrl Z, and you can do cut, copy, etc. Many things you can do. In fact, you can just select any node whenever you select a node, it becomes yellow, and you can just edit from here as well. I'm pressing Ctrl Z to undo the thing I have done already. You can group different nodes together, so on. So these are all just some visualization acrobats with gymnastics, whatever else you can say. You can of course select some nodes and okay, so let's do one thing. Let's select this node and okay, let's select this node, and let me show you what we can do with the select menu option. You can click select, then go to nodes, past neighbors of selected nodes, and select undirected. So what will happen, all the direct neighbors of this node will be selected. So I basically want to okay, it has gone. Let me select again. Select nodes, past neighbors of selected nodes, and then undirected. Okay, now we can do some other gymnastics like rotating. So for that I need to show tool panel. Yeah, you can rotate this thing. See, you can rotate this thing in different angles. You can scale this thing. Scaling, the lower the scale is, the closer they'll become to each other. And align and distribute. So these things are not aligned. So first, suppose these genes are important for some kind of a study, so you want to show them together. So to show them together in a decent manner, we have to distribute them with equal distance. So let's select, let's first align them along the vertical axis. Sorry, this is okay. So all of them are vertically. I mean, all of them are in the same line, same horizontal line. But some are overlapped on something else. So let's just select them and let's distribute them. Horizontal distribute, horizontal distribute center. Okay. So all of them are now more or less same distance from each other in the horizontal axis. Let's select this node and let's select this node and take it somewhere here, so that all of them have some distance between them. Okay, now it looks nice. So all these kinds of gymnastics and acrobat, whatever you say, can be done using Cytoscape. Okay. Now I did not tell you about layout so far, which is an important thing to do. Let's choose organic layout first and see how it lays the network out. It will probably take some time. Okay, so after I selected organic layout, the network looks like this. It's kind of airball, it's still not much readable, but if I zoom in, probably it will not look so dirty. well, it's still dirty. So let's apply some other layout. Can go to Wi-Fi's layouts and select circular, hierarchy, etc. Different selection will give you different layouts. Let's look at hierarchical layout first, and again it is going to take some time. Okay, so I came back to Cytoscape. I had to close Cytoscape because the layout thing was working for more than 1 hour and it did not finish. Even I couldn't cancel, so I closed it and reopened it with loading this high quality.sif network file. So the thing is, we don't have to use different types of layouts. That's a dangerous idea. Actually, I don't recommend it if you have low memory. But the thing is that you can, if you have high convolution machine, then you can use, you can select different types of layouts and see how your network looks until you are satisfied with its look. Okay. So now let's go to some more interesting topics. Let's select tools, network analysis, analyze network. I want to do some kind of network analysis. Okay, so this window comes. I want to treat this network as undirected, so I press OK. Okay. So now it is. I think it came somewhere. Yeah. So this is the results panel that shows the result of network analysis. Let me enlarge it. So here you can see that there are 42 connected components, and actually this is evident from this graph as well. Here is one component, here is another. So there are these are all disconnected from the main connected component, from the largest connected component, and here also there are some other small connected components. So first of all, since we don't want to work with all these connected components, typically what we do, we typically choose the largest connected component, and we can do that very easily by going to tools, network analysis, subnetwork creation, extract connected components, and then select the component which is the largest component. See, there are about 3000 nodes in the largest component, and others have very few nodes. So I'll just extract it. Okay. So as you can see that a new network has been created from the previous network. So this network contains all the components, and this network contains only the largest connected component. But let's finish looking at all the network characteristics we have computed so far. Clustering coefficient is very low. The network diameter is 12. Diameter is the maximum length of a shortest path between two nodes in a graph. And we can see that there are many shortest paths. Characteristics path length is very small, as you can see. So this means this is probably a small world network. Remember about a small world network, which is very low value of characteristic path length as compared to the network size. There are about 3000, more than 3000 nodes, but the average path length, average shortest path length between two nodes is just a little bit more than 4. So this is the characteristics of a small world network. Density is very low. One reason is probably because of the presence of many disconnected modules, disconnected subgraphs. but even if we disconnect them, if we don't consider them, I don't think this will be much higher. Okay, now since this analysis will not give you the correct picture, let us go to the this network, which is the largest connected component of the East PPI network we have seen earlier, and do a network analysis on this network. Okay, here we get it. Now in this network you see that there is just only one connected component because all of the nodes are connected, and still the characteristic function is low. There is no isolated nodes self loops here. Okay, and one another thing I should probably look at here is the node degree distribution. So you can you remember this graph somehow from the slides? What does it say about the network structure? This graph says that our network is scale free, meaning there are very few nodes which have very high degree, and most of the nodes has a smaller degrees and very small degrees. Now we can get a line by pressing this button, fit power law. Okay, so this point fits with this line nicely. So again, this shows that the East PPI network observes power law degree distribution. Okay, let's. There are some other interesting graphs and measures as well, but let's look at some other interesting facts. We can visualize these parameters. So we can actually vary the node size based on different network parameters, like for example degree. So the more connected a node is, it should have larger size. And we can also change the node colors based on some other properties such as clustering coefficient, and then I click apply. As you can see that the graph becomes very nice, and different colors are for varying values of clustering coefficients, and different sizes are for different degrees. Larger sizes are for high degree nodes. Now I want to also look at the colors. Field color, yeah, here it is. So it says that column is clustering coefficient. You can also change the column from here as well. I don't want to change it. Well, I want to keep it clustering coefficient. I may want to change this thing. Double click here, and I want to make the nodes almost yellow color, which are near 0.5. Okay, so this is basically like a cutoff. So I may want to look at only those nodes which is very high clustering coefficient, more than 0.75 or something. So those nodes are present here. Here, all of the nodes have color in the range between yellow and orange. Okay. Now you can probably see which ones have high degree and which ones have high clustering coefficient, but still the relationship between them are not so much clear because they are all of them are embedded into very complex network. So maybe we should look at these types of nodes, like meaning the nodes which have high degree and or clustering coefficient, separately from all other nodes. To do so, I go to Select tab and I want to add some kind of filters. I click plus and I click okay, let's start with degree filter. So I want to select all those nodes which has degree between 100 and 323. Okay, so these are the nodes. Okay. So these yellow nodes are the ones which satisfies this property. Degree of them are between 100 and 323. And if you look at this table panel, you will also see that only those nodes are shown which satisfies the property, meaning has degree between 100 and 323, and you can be sure of that by going to the degree column. Where is the degree column? Here it is, and you can then click on here, then double click here so that it shows the nodes in decreasing order of their degrees. So this is the highest degree node, the first node, which is the highest degree. The highest degree is 284, the next highest degree is 208, so on and so forth, and it is only showing the information about nodes which has degree greater than or equal to 100 and less than or equal to 323. Okay, so now I also want to select those nodes which is high clustering coefficient. But before that, I want to create a new network with these nodes. So remember, these are this can be called hub nodes. Hub nodes are nodes that have high degree. So I want to see whether what are the properties of those nodes. Do they connect with each other as well? And Barabas et al., for example, did not find these characteristics. They found that hubs tend not to connect with each other in PPI and metamolecular networks. So this is a PPI network. So I expect that there is no connection between any two hubs. But is that really true? To verify that, let's go to File, New Network, and select from selected nodes all edges. So that will create a new network. If you go to Network tab, you will see this. This is the new network that has been formed from this network we have been working on. So this new network contains 12 nodes and 7 edges. Of course, there are edges between these nodes, but you can see that these nodes are disassociated, right? So there are very few edges between the hubs. So there are 12 nodes, and with 12 nodes there can be possible like more than 100 edges, but only 7 of them are present. So Barabas's observation was correct, that the hub nodes tend not to connect with each other. There are only very few edges between some of them. Well, let's go back to our previous network, and let's again go to the Select tab here. I just selected those nodes which has degree between 100 and 323. I can apply another filter that chooses nodes based on their clustering coefficients. So let's choose a column. So we can choose any of these columns here, but let's choose clustering coefficient. Now clustering coefficient varies between 0 and 1. The higher it is, remember, the higher it is, the more likely it is to form a cluster with some of its neighbors. So let's look at the nodes which has high clustering coefficient. You cannot see any yellow node here, right? Why? Because probably these two probably are no nodes which satisfy both of these two conditions. Here you can see we are applying an AND logic, meaning we want to select only those nodes which satisfies this condition, meaning the degree should be between 100 and 323. Degree should be at least 100, as well as the clustering coefficient condition, which says that the clustering coefficient should be at least 0.672. Even if we make it 0.5, I don't see any node to be selected here. So what is going on? Let's go to File, New Network, and you can see that this thing is disabled. This submenu is not active. Why? Because we did not find any node that satisfies both these properties. So of course AND will not work. So let's select OR. In other words, we are now selecting nodes that have degree at least 100 and clustering coefficient at least 0.506. And then let's create another network from the nodes we have just filtered. And this network looks like this, and this will be here under the network we have been working. So this network contains both high degree as well as high clustering coefficient nodes, and we can see that the nodes that have high clustering coefficients, meaning the blue ones or bluish ones, tend to form clusters around them, as we have expected. Here. However, that may not be true for hub nodes, which are large sized nodes, and you can see that there are some hub nodes which has very low clustering coefficient, indicated by the rose color. So here is high clustering coefficient, high to mid clustering coefficient nodes form cluster. So we have been talking about clusters from here and there. So it might be a good idea to computationally predict the clusters, meaning the densely connected set of genes. But before doing that, I want to finish with hub genes. What are they? So let's select this hub. So it says this is the name and share. Let's search with this name and see what we can find. So I want to put that name and also say essential gene, because we already know that hubs tend to be essential genes. So I just select this and find this page. I have already found this here. So if I look at this page, I see that this gene which I have just searched, it has an alternative name which is the standard name ABF1, and this gene is actually an essential gene as mentioned here. So that That verifies our assumption that um, gene is probably essential. Now let's select this one. Um, this one is YKL112W, and then select this gene's name, and let's go back here. So I have already searched with this name. I have already searched with this name along with the parts essential gene in Google, and sorry, that was sorry. Let me just restart the whole thing. So this gene was like this was also the same. How about this one? Okay, the shared. No, I should. Let's select this hub. It has also a very large size. So how about this? Is it also a shared gene? Okay, this is RAP1. Okay, it says at the very beginning, essential DNA binding transcriptional regulator. So this is also an essential gene. These two have the same name, so I don't want to check those ones. I may check this one, for example. What is this? YNL. Okay, I think I just did search for this YNL 216. How about this one? YBR049C. This is REB1 gene. REB1 is the standard name. Is it also essential? Let's see. Yeah, this is also essential for a particular strain of Baker's yeast. So in this fashion, you can see that hubs tend to be essential as observed by earlier researchers as well. So far, we have made a number of observations. That is, high quality PPA network has these properties. It's a small world, as we have seen from the where is my results panel? Somewhere here. Okay. So when we have looked at the characteristics path length, we have found that it's only 4 point something. So that's why it is a small word. So let me just write it down specifically. Characteristics path length equal to 4.23, while network size is much larger, almost 2000, almost 3000, 2924. So that's why it is a small world. Scale free, we have seen why it is a scale free. So let's look at the result panel again and see. So we can actually export this thing as an image, and let's save it as somewhere maybe in the desktop. Scale free, and let's select JPEG, save. Okay, And let's open this using Microsoft Paint, copy it and paste it in our document as a proof that this is really a scale frame. Meaning if we put the degree in the x-axis and number of nodes with that degree in the y-axis, we get a distribution like this. Okay. Also, we have seen that halves tend to be disassociative, meaning that halves tend not to connect with each other, and that is verified from this figure, which contains only the halves. There are only 7 edges between 12 halves. Only 7 edges between 12 top 12 halves. And also we have observed that halves tend to have low clustering coefficients. Halves tend to have low clustering coefficients. That's why we did not find any when we worked on this network, this largest component network. After applying the AND logic, we could not find any node that satisfies both degree property and clustering coefficient property. That satisfies that requirement that both degree and the clustering coefficient should be high. We did not find any such node which is degree greater than 100 and clustering coefficient greater than 0.506. There is no node which has degree greater than or equal to 100 and clustering coefficient 0.56. And halves are also tend to be essential genes. Actually, not as essential genes, just essential genes. That was a mistake. So, so far the halves we have checked, they satisfies this property, and that includes REB1, TAB1. REB1, TAB1. You can check the rest. Okay, now let's go back to Cytoscape and let's go back to our original largest component network. And now the other thing I want to show you is what else we can do with network analyzer. Select network analysis, we have seen how it works. Analyze network, and after I analyze network, I get a ledger panel like this, and it has some tabs which I can see. Also, you see that these kinds of things can be exported as well. You can export this data for each of these tabs. You can export this data and then load it. And for loading, we have to go to Tools Network Analyzer, load network statistics. We can also analyze subsets of nodes. After selecting some nodes, we can analyze only those nodes. But whenever we do this, I find that even if I select only a few nodes, it takes a long time, and so I don't recommend it, especially have low memory. Batch analysis is for analyzing multiple networks that are present in the same directory, same folder. So, for example, we can select the directory that we have selected earlier by going to C, program files, and then Cytoscape, and we can select sample data. We can select this folder. So all the networks of this directory will be analyzed, and we have to of course select another directory for creating the output files. And after selecting some option, we can start analyze. And if we do so, all the networks in this directory will be analyzed, but it will of course take a lot of time, so I'm not going to do this now. We can plot parameters. So for example, we can see how the degree versus clustering coefficient plot look like. So x-axis degree, y-axis clustering coefficient. So you can see that most of the nodes has low degree, and those low degree nodes have a varying values of clustering coefficients. Okay. Generate a style from a statistics. Again, you can vary node size and node color based on some graph property like degree, clustering coefficient between separate, etc., etc. We have seen this thing. We have seen subnetwork creation also, how to extract connected components. Okay, so these are the main things in network analyzer. Of course, we can do many interesting things with network analyzer. We cannot compute clusters, for example. So for that, we need to install some app. There are different apps available in this website. If you go, if you just search for Cytoscape app, you will find this link, and it contains many interesting apps. Some of them are for analyzing networks, and some of them are for clustering. So cluster one, as I have told you, is a recent clustering algorithm which shows very good performance, and also M4 is an old one, but it is a good one as well. So I have installed cluster one by just clicking here and then clicking install button here. It takes just a few seconds. So I have already installed it, so it is available in the apps section. If you didn't install it yet, so install it please. So now if you go to apps, you'll see a menu for cluster one. You just click a start. Now there are different parameters you can set. I have already set minimum density of 0.4 and minimum size is 3. 3 is too low. If you give 3, then it may result in a spurious cluster. So let's make it say 20. Okay. So these are different cutoffs we want to apply on the clusters it is going to compute. So you can see that cluster 1 app has already calculated some kind of clusters which are shown in red here. All other nodes have changed colors, and the results are available in the results panel, which is here, cluster 1 results. So there are just 3 clusters that satisfy our stringent conditions, that the number of nodes should be at least 20 and density should be at least 4. This cluster does not seem to satisfy the density cutoff, but it is close to 0.4, so it is okay, I guess. So we can look at these clusters and see what those genes correspond to. For example, these clusters, I want to embed this here for my convenience. So there are this cluster, yellow cluster, and these clusters as well. Okay, so whenever I select a cluster here, that is selected here, it becomes yellow. Okay, so it has 38 nodes. which are shown here. Now we can again go and look at what those genes do, if there is something common among those genes. Typically, genes in a certain cluster tend to have some common function, or they tend to participate in the common protein complex. So for that, we need another kind of analysis, which is beyond the scope of this workshop. For that, we need functional enrichment analysis. So I'm not gonna talk about that right